Good evening, everyone. I hope I am audible to every one of you. A very good evening and warm welcome to every one of you for joining this webinar once again today for paper discussion CSIR NET exam paper discussion 2021 Unit 5. Before that, before we proceed with the session, everyone, please confirm that I am loud and clear and you can see the screen. Come on. Just write yes. Okay. Good evening, Devanshi. Good evening, Kirtana. Nice to meet you all once again today in this paper discussion session. I hope you are really enjoying the session. It has been discussed till Unit 4. So today we will be taking up Unit 5. So Unit 5, we all know it's all about developmental biology. And it has got two sections. It has got animal developmental biology and plant developmental biology. So first, I will be taking up plant developmental biology. I will be discussing questions that have come from this particular part in your examination, and questions from animal developmental biology will be taken up by Mrs. Urmimala ma'am. All right, so let's get started. So this time, yes, we had a very good and nice set of questions that had come from this particular part in both the shifts. So let's begin. So first, I will be taking up Unit 5, Shift 1, CSIR question paper analysis, and we'll begin with Part B questions of Shift 1. So let's start. So coming to the first question, which of the following combinations would best describe the dominant phase of a life cycle of a pteridophyte? So every time when I also discuss these concepts in class, I always say that a plant life cycle would be having a dominant and a recessive phase. Not a recessive actually in that term, but still it has got a sporophytic phase and it has got a gametophytic phase. Okay, sporophytic and gametophytic phase. So we normally say that in angiosperms or in phanerogams, the sporophytic phase is dominant and gametophytic phase depends on the sporophyte for its nutrition. So when we get to see a plant or a tree, okay, when we peep outside of our window to see a plant, so which exactly the part we are viewing? It is definitely the sporophytic part we are viewing because it's the dominant phase. And when it comes for the cryptogams, which are considered as the lower group of organisms, for them, we generally consider that for them, the gametophytic phase is dominant and sporophytic phase depends on gametophyte. Why so? Because normally the cryptogams do not have a shoot root differentiation, right? And we get to see they have a compact thalloid structure and they have reproductive structures on it. So the reproductive structures are very much predominant. And hence, the gametophytic phase is definitely dominant for them. So now, based on that concept, we have a question here: which of the following combinations would best characterize the dominant phase of life cycle of a pteridophyte? And which of the following is the correct option? So will it be a diploid gametophyte, haploid gametophyte, diploid sporophyte, or haploid sporophyte? So come on, any guesses? And mind it, pteridophyte, although it comes under cryptogam, but though it's a complex plant. Okay, pteridophytes were the first vascular land plants, and they do have a proper root shoot differentiation. Okay, so based on that, any guesses? Did you attempt this question? Let me see. Yes, Selva Priya, very good evening. So, can I get some answers from your end? Okay, Selva is saying C, okay. Others, Kirtana is also saying C, okay. So, let's see. Okay, that's wonderful. Yes, it's option 3 or answer key 215. So dominant phase of pteridophyte is the sporophyte. Yes, why it is so? Because you might have this question, why it is for pteridophyte? Because yes, exactly, Ashwin, it's C or option 3, because pteridophyte is a complex land plant. Okay, so it's not really having the features that a typical cryptogam bear. So meiosis occurs within sporangia located on the underside of the sporophyte leaf. And after the spores are released, they germinate, they divide by mitosis and grow into simple heart-shaped gametophytes. Okay. And on the gametophyte, we have cells in the archegonium and theridium form the eggs and sperm, and the sperm swim to the egg and fertilize it. So that means what is option C? Diploid sporophyte. Okay, so as I was saying you, gametophytic structure means archegonium and antheridium are the reproductive organs. So they will be the, not dominant for sporophytic phase. Correct? So definitely whenever, if it would have been bryophyte or algae, or that is thallophytes, then definitely the answer would have been gametophyte, haploid gametophyte. But as because it's pteridophyte, hence the answer should be diploid sporophyte. Okay? Now, next question. In which of the following stages of Arabidopsis embryogenesis do the visible distinctions between the adaxial and abaxial tissues of the cotyledons become initially apparent? Okay, first of all, students, you need to understand the question, what has been asked to you. That is very, very important. So, in which of the following stages? So, we all know about plant embryogenesis, correct? And plant embryogenesis have different stages of it, starting from the, zygote, then diode, then quadrate stage. After diode, we have quadrate, we have octant, that is 8-celled, which is globular stage. Then we have heart stage, we have torpedo stage, and finally, we have the maturation stage. So the question that has been asked here is, in which of the following stages of Arabidopsis embryogenesis do the visible distinctions between adaxial and abaxial tissues of the cotyledons become initially apparent? So adaxial and abaxial. Adaxial means upper surface. Abaxial means the lower surface. So in which particular stage of this Arabidopsis development we get a better clarity of this upper and lower surfaces? So let's check out the answer. It's option 3 once again. That is diploid sporophyte. Yes. So Arabidopsis again is a diploid plant. So whenever, and it's definitely considered to be a phanerogram. So it's not a cryptogram. And again, whenever we are considering Arabidopsis embryogenesis, we have to think about it that adaxial abaxial tissues become or will be prominent in that sporophytic stage only, but not before that. Okay. So wild type, so the explanation for this goes as, wild type leaves are characterized by anatomical differences between their adaxial, that is upper surface, that I told you, and abaxial means the lower surface. So the adaxial side comprise of dense columnar palisade mesophyll, where the abaxial side comprise of spongy mesophyll cells. And at the mid torpedo stage, so torpedo is almost the towards the end of embryogenesis, because it's the 32 cell stage is generally considered to be as the torpedo stage, we all know, because right after torpedo we have maturation stage. Okay, so towards the mid torpedo, enlargement of cotyledons, hypocotyle, etc., come out, and at bent cotyledon stage, embryo with elaborated radial pattern in different organs take place. So, in the cotyledons, a single adaxial subepidermal layer of elongated cells can be distinguished from the underlying mesophyll cells. Okay, so that is what. So, in this question also, we have to understand the correct option should be a diploid sporophyte. Now, coming to the part C questions. Okay, yes. So, this was a very interesting question, everyone. Following are certain statements regarding root growth and differentiation in plants. So, what are the options given here? Root hair, endodermis, xylem, phloem reach maturation in the elongation zone of a developing root. The root epidermal cells that are incapable of forming root hairs are called etricoplasts. So, this is a very direct statement that they have given this time. So, etricoblasts, yes, exactly, it is considered to be like that when these root epidermal cells, they are not capable of forming root hairs anymore, then those underdeveloped I would say, would be root hairs will be called as etricoplasts. Now, quiescence center is present just above the root cap. Absolutely. So that means, are you able to follow that whatever statements has been given here, students, it is absolutely based on a very basic knowledge. So we have to be very much clear about that. What is a quiescence center? Where it is present for the root epidermal cells. So always, or normally, what we focus is on the shoot apical meristem or SAM concept, but we normally don't focus on the RAM. So yes, we now have to focus on root apical meristem as well, because the question pattern is saying that normally CSIR till date, they did not give questions normally from this RAM, but now they have started giving questions from this part. So we have to prepare well for RAM as well. Different genetic factors that control root apical meristem, that also. And then Arabidopsis and auxin transporter ABCB4. Okay, ABCB is a type of a transporter. It helps in the transportation of different hormones also. Like ABCB is a very important transporter of auxin. It plays a role in root hair emergence by maintaining intracellular auxin concentration. So students, can you guess that which can be the correct option? Yes, Shubhash is saying A. Okay. Others? So let us see. So the zone of elongation is where the newly formed cells increase in length, okay, thereby lengthening the root. So no maturation take place in elongation zone. That is why the statement A is incorrect, I'm sorry. That means root hair, endodermis, these are the matured parts of a root. Correct, students? And it has been said here that they are reaching the maturation or reaching maturity in elongation zone of a developing root. But just come to the reasoning part. Zone of elongation is where newly formed cells are being added up. Why? So that it helps in increasing the increase in length or lengthening the root. So no incident or no events of maturation will be taking place in the elongation zone. That is why statement A is incorrect, and definitely the other combinations are absolutely correct. B, C, D. That means, hence, option 2 is correct. Yes, Bhagyashri, you're correct. All right. I hope this is clear to everyone. Now I'll proceed. So yes, next is Unit 5, shift to CSIR question paper analysis that came from Plant Developmental Biology. So let's check out with, I guess here only part B question was there from Plant Dev Bio, because no, the weightage is more for Animal Dev Bio rather than Plant Dev Bio. Okay, if total 8 questions supposedly or 10 questions are coming from no entire unit 5, so from part B we can expect at least 3 from animal or 2 from plant, 4 from animal, 1 from plant. So this is the normal ratio that comes for plant developmental biology. Okay, so which one of the following transcription factors is important for delimiting the meristematic and elongation zones of the roots? It's a very direct question, and I would say, students, follow Thays and Zager. Okay, 5th edition for plant developmental biology. Whatever genes, the genes that you are seeing here in the options, okay, each and every gene is there. They're functional, their functions, everything has been mentioned. So I always suggest for this part, specifically for plant embryogenesis students, try to focus, try to get hold of Thesenzeger 5th edition, and the chapter there is growth and development. That is the chapter for plant and development. You'll not get the chapter name directly as plant embryogenesis. Okay. So which one should be the option? It's a very direct question, and you have to be very thorough with each and every function of the genes mentioned in the book. And the option is option 3, that is plethora gene. Plethora, it comes in 2 genetic products, PLT1 and PLT2. So apart from this delimiting the meristematic and elongation zones, PLT also control the auxin concentration as well. Okay, so it has got a relation with auxin regulation as well. And everyone knows that how auxin is important for root development, right? Yes. So it's a Brief description here given. Members of this family, Antegumenta, like this is a very important family of proteins, floral organ proteins. Okay, and the PLTs are expressed in the embryonic and adult root meristem, overlapping in their expression domains, mainly in the quiescent center. Okay, and I'm not going into the other details. Yes, we know that how short root and scarecrow genes are also important. We know that they help in developing the radial patterning to take place. What is radial patterning? Development of the different tissue layers like ground layer, formation, vascular tissue formation. So SHR and scarecrow have a specific role in these. Okay, so that is the thing we have to focus on this, that plethora gene has got a very important role in delimiting the meristematic and elongation zones of roots. Okay, so that's all, students. We had only this 4 to 5 total number of questions that came from plant developmental biology. And next, the animal developmental biology question paper discussion will be taken up by Urmimala ma'am. So over to you, ma'am. Thank you so much, everyone.
Am I audible to you? Please confirm in chat box if my PPT is visible and I'm audible to you. So today we are going to see the second part of development biology, which is animal dev. You just saw the questions from plant development biology. Let's check from the animal dev bio. Okay. All right. So your first question from shift 1, that was about amphibian gastrulation. Yes, very good evening to all of you. Yes. So gastrulation begins with the invagination of bottle cells, followed by coordination and involution, and last will be epiboly of the ectoderm. Okay, involution of mesoderm and epiboly of ectoderm. So you have to say which out of the statements is correct regarding amphibian development. Okay. So organizer induces the nucleus center. Organizer is formed by accumulation of beta catenin. And in the absence of BMP inhibitors, the ectodermal cells will form neural tube. Now, which out of this statement is correct? You have to select any one of them. Did you attend any of these? Can you take a guess which out of these options will be correct? Amphibian development is a very important topic from animal development biology, and every most more or less every time the questions come. Yeah, very good, Selva Priya. That's the right answer. Okay, statement 1 is the right answer because it's a pretty simple thing about gastrulation. Forget about the other ones, but if you know this concept that yes, it starts with a formation of blastopore. This invagination of bottle cell what creates a blastopore in the entire blastula of frogs. Because of that, an invagination is started. Okay, so that is what is the blast. Yes, correct. Bhavya, Devanshi, Vijay, very good, all of you. Correct answer you've got. Followed by coordinated involution of mesodermal precursor. Now let me show you the diagram. Yes, the answer key was 1. So vegetal rotation and invagination of bottle cells. So this is kind of invagination you get. Can you see a lip of blastopore created, right? And if you see here the entire thing, now this concept will be required later also, because one question was asked in shift 2 in part C regarding this. Okay, so as you can see over here, this is the fate map, some colorful diagrams with what they give, right? So we know that this is a fate map. Fate map means in early embryo we will be marking some parts in the embryo itself, that what they are going to form later. Yes, Bhagyashri also correct, Bhavya right. So what they are going to form later, we will be drawing something, right? Now here you can see this is the dorsal side and this is the ventral side. So this part is called as marginal zone. This part is the marginal zone, and there are some regions in the inner marginal zone which are towards this side and outer marginal zone. So what parts are there in inner marginal zone? Head mesoderm, the ones which will form head mesoderm. There a molecule called as goosecoid is expressed. Okay, if I just show you over here. So as you can see, this part will be forming head mesoderm where the goosecoid is expressed. Okay, and this part is going to form chorda mesoderm later, which is expressing XBRA. So we will be talking about this later. Remember it. Okay, this part is XBRA and this part is goosecoid, and you can see that here the blastopore formation will be taking place. The blastopore means what? An opening. So where will the opening take place? You can see the bottle cells, how they are going inside. They have a change in the shape. So because of the change in the shape, do you notice a pore is getting formed? Right? So that is what happens in the first place. Internal cell arrangement propel the cells of dorsal floor of blastocele towards animal cap. This places the prospective endoderm cells adjacent to blastocele and involuting mesoderm. So when that happens, you can see the red color cells, those are the prospective mesoderm. Generally in fate map, we have a particular color. That blue color is marked for ectoderm. As you can see, they will form later ectoderm. Prospective means they will form later. They haven't formed yet because this is early embryo, no? That's why. Red color we will put for mesoderm and yellow color we will put for endoderm. So these are the color markings in your reference standard reference book, that is Gilbert, which we refer for development biology mostly. So as you can see, the mesoderms are going inside, and that mesoderm going inside is what is called as the involution. So as you can see, involuting mesoderm, and these together it creates a blastopore, right? So cells will then move, and all that involution of the lip will take place after the bottle cells have brought the involuting marginal into contact. The inner marginal zones also will involute into the embryo. That means XBRA goosecoid, they will also now involute. So as you can see, those orange, light pink color, red color, see light pink color, red color, light pink color, red color, light pink color, red color. So can you see out? They were outside, but later you can see slowly they are moving inside. So finally they are inside. This is involution when they go like this. You follow the hand movement, they go like this. Okay. Invagination is simply lifting of a membrane, so that a layer of cells, so that a pore is created. But involution is moving like this. Okay. Around a certain pore, they are moving. And last topic, what we have is the epiboly. Okay, so epiboly, what happens? You can see outside it is showing ectoderm begins epiboly. Can you see this blue color part, how it is coming and trying to enclose? Earlier the blue color part was still here, only till here. So blue color part basically means where it will be trying to form the like ectoderm. Okay, so it has to spread like this. So that's what happens during frog gastrulation. Bottle cells of the margin move inward through invagination to form the dorsal lip of blastopore. Mesodermal precursors involute under the roof of the blastocele, and towards the end of gastrulation, embryo becomes surrounded by ectoderm. So ectoderm is internalized and mesodermal cells are positioned between ectoderm and endoderm. At the end of gastrulation, you have an outer layer of ectoderm, a middle layer of endoderm, and in between them you will have mesoderm. So that's why we can say A is the correct answer. So if A is correct, obviously the others go wrong because only one statement you have to choose correctly. But still, I'll tell you what happens. It is saying that the organizer induces the nucleus center, but it is the other way around. Actually, nucleus center induces the organizer. Organizer is formed by the accumulation of beta catenin. No, accumulation of beta catenin creates the nucleus center, and the nucleus center is going to induce organizer by some gene expressions, especially the goosecoid. Okay, and in the absence of BMP inhibitors, so wherever the new organizer will be there, that organizer is going to synthesize BMP inhibitors. Okay, and wherever BMP inhibitors are there, that side neurulation will be taking place because BMP inhibitors are at the dorsal side. So that side neural tube will form. But what it is saying, in the absence of BMP inhibitors, the ectodermal cells will form neural tube. How is that possible? Obviously, if BMP inhibitors are not there, they were supposed to cause dorsal. If they are not there, then how can dorsal structure form? Not possible, right? So in that case, ectodermal cells will go for ventral phenotype. Okay. So the dorsal most vegetal cells called the nucleus center. So as you can see, this is the nucleus center. You can see nucleus center is sending some signals to the top. Because of that, that area has formed Spearman organizer. Okay. So it's the other way around. So B is wrong. And major candidate for the factor that forms Newcoop center in these cells, it is beta catenin. Okay. So can you see the accumulation of beta catenin? Because of that, some genes were expressed, and that indirectly led to formation of Newcoop center. Newcoop center formed organizer. So even here it's wrong because it directly says organizer formation. And BMP inhibitors, they form organizer, means they are formed from organizer tissue. They block the formation of epidermis. Actually, this ectoderm can have two fates, right? So it can have a ventral fate, it can have dorsal fate. If it is dorsal fate, it will go for neural tube. If it is ventral fate, it will go for the epidermis formation, right? So these BMP inhibitors at the dorsal side, they will not allow the formation of epidermis. So here they will form neural tube. But if BMP inhibitor is not there, then here also it will form epidermis, right? So in absence of that, it will not form neural tube. That's why the rest are incorrect. Next question from part B. So this is about development of sea urchin based on beta catenin accumulation. So they are saying that beta catenin prime predominantly is expressed in the micromeres. So you know that in the sea urchin, you have several type of cells. You have some mesomere cells at the animal side. Then you have some big cells. Those are the macromeres. And then you have some very small cells, which are the micromeres. So there are different tiers of cells. Tiers means layers of cells. Okay. So they are saying something about this micromeres, that beta catenin accumulates over here, right? And these cells, they will be fated to become endoderm and mesoderm. Okay, because these cells are going to give signals to the up cells, the cells above them. So they are asking, if GSK3 is blocked in these embryos, then what is going to happen? Which out of the following will happen? So again, you have to select only one statement. Can you tell me this? Okay, let's see. Selvapriya, we will see. Let's check. Okay, Ashwin also have given answer. So yes, both of you are right. Okay, the correct option is indeed answer 3. Now, what have they told you? Something about beta catenin. Okay, first, something about GSK. So what they have said, if GSK is blocked, so we have to consider a condition where GSK is blocked, means not there. So Bhavya, what you can do is, no, see there is a setting, no, on the top. So just click on that setting, okay, and you can change the resolution over there. If you click on the setting, then the quality you can select from there, either 720p or 1080p, whatever is suitable for you. Then you will get proper visualization of that. Okay, just try it. Okay, so we have to understand that GSK is blocked over here. Now there are three conditions given. This is the wild type condition. So what happens in wild type? See, this is a part of a signaling process. There is something which is called as a WNT signal. Probably you will know that. So WNT signal, when it comes inside the cell, there is a receptor which is called as frizzled. Frizzled will activate a molecule which is called as disheveled. Disheveled will inhibit. You know the sign for inhibition, right? It will block GSK complex. And GSK complex was blocking beta cat. But if GSK complex itself is blocked, then beta cat will be free. So basically it's a double inhibition. So double inhibition means this should be available to block this. But if this is blocked, then beta catenin in that case will be free, right? So we have to understand that if GSK is blocked, our beta cat will be available. And it was said in the embryo. So in all cells of the embryo, whatever cells we had there, in all the cells it is expressed. Okay, that's what we have to understand in the experiment. In this experiment, we have to understand. Okay, but usually beta cat is only expressed in the micromeres. That also you should know. It is expressed only in the micromeres, the beta catenin as such. But in this experiment, our beta catenin, so this is wild type. Can you see here? This orange molecules means these are the nucleus. And beta catenin being free, because in the micromeres they are free. Naturally they are free, so they can enter the nucleus, and that is shown here by orange color. Okay, and somewhat less in the large micromeres, somewhat less. Small micromeres and large micromeres less, but small micromeres maximum. But you can see the rest of the embryo doesn't have beta catenin, right? But now we have a special treatment by lithium chloride, that is experiment 1. So what happens if you have treatment with lithium chloride? So GSK is supposed to block beta catenin, but then lithium chloride will block GSK. Got it? So lithium chloride will block GSK, and because of that, beta catenin will be free. As I told you, if GSK itself is blocked, generally by this signal it is blocked. By WNT signaling, the GSK is blocked. But that is happening naturally, it is happening in the micromeres. This process is naturally taking place by signaling in the micromeres through WNT signaling. But other cells, they don't have this process. So what happens? Their GSK will be present. Okay, in all these cells that signal is not there. So GSK will be there. But what have we done? We have treated the entire embryo. We have treated the entire embryo with lithium chloride. And lithium chloride is blocking GSK all over, not just by WNT signaling in micromere, but here also GSK will be blocked. They were supposed to be present, but they will be blocked. As a result, what will happen? Now these also will express beta catenin. So can you see the orange color? Now beta catenin is free in these cells also. Now question is what this beta catenin presence was doing. So if you can see the wild type, it is a larva, Plutus larva, which is formed in the sea urchin properly, where outer layer of ectoderm is there, inner layer of mesoderm and endoderm is there. Everything has happened properly. Okay. But lithium chloride treatment permits beta catenin to be free and accumulate in all blastula cells. Okay. Leading to the animal cells becoming specified as endoderm and mesoderm. So as a result, what happens? This is ectopic beta catenin expression. Ectopic means additionally somewhere else also it will be expressed. They were supposed to be expressed only here, but later they are expressed everywhere. So what will happen? Initially, what was supposed to become? The micromeres were supposed to become endoderm and mesoderm. And the rest of the cells, they were supposed to become ectoderm, right? But now what will happen? Even the animal cells, these are animal cells, even instead of becoming ectoderm, because they also have now beta catenin, even they will form endoderm and mesoderm. So all cells are forming endoderm and mesoderm, no ectoderm. That's why let's now check with the options over here. Beta accumulation in the nuclei of large micromeres will be inhibited, leading to formation of ectodermal ball. Why so? No, because it won't be inhibited. Because anyhow in the micromeres, beta catenin was present by normal way. That is WNT signaling. Normal way it was present. Okay, by WNT signaling. So this won't be inhibited. Beta catenin will accumulate in the nuclei of all blastula cells, leading to the formation of ectodermal ball. So even here also, the beta catenin is supposed to accumulate in nucleus of all blastula cells. That's correct. Till here it's correct. But it won't form ectodermal, because if beta catenin is not there, then it will form ectoderm. Okay, but beta catenin is present now even in animal cells. So that's why it will lead to animal cells getting specified as endoderm and mesoderm. That's why it is correct. Beta catenin, which accumulate in the nuclei of large micromeres, will be inhibited, leading to animal cells getting specified as endoderm and mesoderm. Okay, so they will get inhibited by getting specified. That is also wrong, because it will not accumulate in, already it was accumulated in large micromeres. They won't be inhibited over here. So no question. Option 3 is the right answer. Good. Those who got it correct. Okay. So during normal development, it will accumulate in those cells that is in the micromeres, which are fated to become endoderm and mesoderm. And that is autonomous. But what you have done is an experiment by lithium chloride treatment which blocks the GSK. That is experimental. This is autonomous. This is wild type. This is autonomous. You have to understand where it is naturally expressed and where it is made to express by experiment. Okay, then only you will be able to analyze. Another question from Animal Dev in Shift 1, Part B. In transplantation experiment, the area of presumptive ectoderm from a frog gastrula, you have taken from early frog gastrula, the part which is supposed to form ectoderm, and you have taken that part from frog, you have taken and you have put into a nude gastrula. Both are amphibians only, but different species. So you have put it in the nude gastrula, where you have put the part where it is supposed to form some mouth parts. Now, some resulting, you got some salamander larva. So, what did it have? It had frog like mouth parts. Because newt, it is supposed to form balancers. But because you had put frog tissue in the newt, which was supposed to form ectoderm, so instead of balancers, it formed frog suckers. Frogs have suckers. So, why this observation was made? That instead of balancers, it formed suckers. Okay. So, this is an example of what? Determination, genetic specificity of interaction, regional specificity of interaction, or autonomous specification. Which out of this is correct? Yeah, Vijay. So, are you suggesting option 2? Okay, I'll tell you, let others also answer. Yes, what was the option you selected? Others? Okay, Vijay, we will see that whether it's correct or not. So basically, some signals are getting received, isn't it? Some induction is happening. Because of that, the balancers or whatever, something is getting formed over here. Yes, absolutely right. Okay, so both of you are correct. Vijay and Bhavya, both of you are right. Yes, it is genetic specificity. Because simple logic, students, you have transplanted a part of frog and you have put it into a newt. So, how will the frog tissues, how will the frog cells know what to form in the newt? Okay, they have some area that, okay, they have been transplanted in the part where they will form mouth. So, they know they have to form some mouth part according to the signals that is it is getting from the nearby cells. But what type of mouth it will form? The one that it is supposed to, that it knows how to form. Yeah, Selva Priya also right. Very good. The type of mouth it knows how to form. Otherwise, how will it know to form newt parts? Isn't it? So, yeah, okay, very good, Vijay. You got this correct. That's right. So, we had discussed it in classes, right? So, resulting salamander larva had frog like mouth parts instead of balancers. So, this is genetic specificity. A property of epithelial mesenchymal interaction is genetic specificity, whereas the mesenchyme may instruct the epithelium as to what sets of genes to activate. So, this was the experiment actually. From frog gastrula, you took a part, put it in newt, and now you see the newt looks bit different. This is not how the newt should look. So, it is having part of the frog in its mouth area. Okay, so in this mentioned experiment, the mesenchymal cells instructed the ectoderm to make mouth. Yes. See, you have transplanted what tissue? Ectoderm. Okay, but ectoderm tissues changed their specification. So instead of forming some epidermal layers or anything, it formed mesodermal tissue. That is the mouth tissues it formed. Okay, so it changed its specification. but it will only change the type of mouth it knows what to form, because they have different genes, as simple as that. So ectoderm responded by making only the kind of mouth it knew how to make, no matter how inappropriate. Whereas the other type, which was there, one of the options, regional specificity. Example of regional specificity is where chick epidermis secretes proteins that signal underlying dermal cells. So you can see here there is a wing epidermal epithelium in chick, and these are all sources of dermal mesenchymal cells, that is wing, thigh, foot. So different mesenchyme is sending different signals to the same tissue, and the same tissue is responding to different signals and making different parts. That is regional specification. What region it is getting signal, according to that it is responding. So definitely it is not regional. And why A and D is wrong? Because if this was determination or autonomous specification, according to the experiment, if it was autonomous means it will, whatever it has got the specification, it is because of some cytoplasmic factors inside it, and it will always go for that specification. It will not change its specification at all. And same logic for determination. A determined cell is at the last stage of specification, where it cannot change its specificity even if it gets any other signal. So they also won't be correct, because we are seeing a certain change in their specification. Okay. So correct answer is option 2. Good. Next, now this is from shift 2. Part B questions only from shift 2. What would be the effect of retinoic acid treatment on the positional information of blastema cells present on the amputated newt limb? Question from Regeneration. So basically you have a frog limb, okay, a frog limb you had, but you amputated it. And in that amputated area, you have put retinoic acid. So based on that, they're asking what will be the effect. Always when you get a question like this, try to draw a rough diagram. I'm sure even in online exam you will be provided with a sketch paper, the rough paper or pen. So always try to sketch a diagram. It will help you recollect things better. Okay. What would be the effect of retinoic acid treatment on the positional information of the blastema cells present on the amputated newt limb? Okay, so what the answers are, which after this option is correct? You have to select any one of the correct statement. So can you answer this question? No one attempted this? Not sure about regeneration? Okay, so see here, retinoic acid administration changes the positional information in a proximal direction. That you should do, so that a complete limb can be regenerated from a hand. So you can see the diagram, this is a normal regeneration. So if you are cutting it here, if you are amputating here, arm amputation, so you can see there is a arm replacement, that wherever you have cut, only the part which was missing is developed. Similarly, if you cut over here, only the hand amputation you are doing. So you are cutting it only here, only what part is missing, that is developed. You don't see any extra hands getting developed or extra parts, okay? But see what happens if you cut it over here. So you're expecting this much part to be degenerated, but you have treated with retinoic acid. So when you treat with that retinoic acid, you see the entire one starting from the proximal part is generated. Proximal means closer to the body. So wherever this is the rest of the body, right? So these parts will be proximal. These parts will be distal. Basically, retinoic acid changes the position information. If you don't put any RA, then basically if you cut here, automatically the cells get some signals of regeneration, and they get to know that, okay, from here it is the hand part only that will generate. But when you put retinoic acid here, the cell signals will think that, okay, this is the proximal part of the body. So automatically they will start from forming from the proximal part. Okay. Distal. So basically this is a distal part. So only distal part is supposed to form. But when treated with retinoic acid, it's a derivative of vitamin A, regenerate an entire arm composed of all three limb segments. So this respecification of distal to a proximal is seen in axolotl limbs. Axolotl is also a type of amphibian and found to be dependent on both concentration of RA and also how much length you are treating it with. So basically, remember, it induces proximalization of regenerating tissues and duplicates limb elements along the proximal distal axis. Following amputation, the regenerate is identical to the tissue which is lost. If it is treated with retinoic acid, then more limbs and more tissue is regenerated and limb elements are duplicated. Okay. So yeah, the RA will have no effect. No, it will have effect. Respecified to more distal? No. Respecified to more proximal. Cells will lose their positional information. That's correct, but they will remain as dedifferentiated cells. No, they will regenerate a proper arm. Okay, that means it will respecify itself. Okay, next question. Again, from shift to a part B question. Very easy question. The embryonic stem cells in mammals are derived from what? That was the question. Extremely easy. I am sure most of you would have attended this question. So if not, then you are having some issue in figuring out or just finding the questions from the exam paper. You should develop this capacity to locate the questions from question paper. Okay. So immediately when you look at this question, sometimes these are hidden and you may not be getting it. What, students? No answer for this? This is the easiest question. Embryonic stem cells in mammals. Where do you get it from? So, mammals, I hope you know that there are some layers of cells, right? So, the outside layer of cells, the blastula, what is formed? Blastula means a group of, means a collection of cells, right? The embryo which is having the collection of cells. Okay, Kirtana? Okay, you've given an answer. And there are some cells which are present towards the inside part. So inside part cells are called inner cell mass. The outside cells are called trophoectoderm. Okay, since it's outside, no, ecto part means outside, so trophoectoderm. And this empty part, what you see, that is the blastocyst. So basically, you need to know stem cell. Yes, very good. Kirtana, Vijay, Devanshi, all of you are right. So basically, you need to know what is the potency. Inner cell masses, they are pluripotent. Okay, they will not be able to form placenta, but they will be able to form any other tissues. Trophoectoderm will form extraembryonic membranes, that is placental parts. So basically, embryonic stem cells, which will be able to form any other kind of tissue, they come from inner cell mass. So correct answer is option 2. Embryonic stem cells are immortal stem cells that own multilineage differentiation potential. They are commonly derived from inner cell mass of pre-implantation embryos. Okay, so this is the diagram. This is blastocyst. This is from a fertilized egg. If you take, right, you can make some changes also later. So this is the blastocyst, this is the inner cell mass, this is trophoectoderm outside layer. If you take the inner cell mass and culture them, you're basically having embryonic stem cell culture. Then you can make some changes and you can once again put them back. So some transgenic or cloning if you want to do, these will be useful. They are commonly derived from inner cell mass of pre-implantation embryos. Another part B question, this was also a very, very easy question. Movement of epithelial sheet spreading as a unit to enclose deeper layers of embryo is termed as what? So these were one of the movements of gastrulation. Hopefully you know all these movements of gastrulation. These were all discussed in Biotecnika classes. So can you tell me which will be the answer over here? Epithelial sheet spreading as a unit. Yeah. So basically you need to know as a unit. So not only as a unit, Vijay, you have to also see that it is enclosing deeper layers of embryo. Means it is spreading. Okay. So yes, I did show you the process of involution before. Could I see the previous question? Yeah, Ashwin. So here is the previous question. Embryonic cells are derived from what? Blastocele, inner cell mass, trophoectoderm and trophoendoderm. Okay. By the way, if you join our Telegram group, there the PDF of this question paper and answers will already be there. So you can refer over there also. Okay. If you join our Telegram group, then you will, excuse me, find the PDF of the question paper and answer. Okay, Kirtana has said involution. All of you are saying involution. Why so? This is epiboly. Okay, they will enclose deeper layers. Involution is generally they will go inside. So remember I told you in the blastocyst, this amphibian blastula I told you. So the mesodermal cells are involuting inside like this. They are going from outside through a pore and they are going inside. That is involution. Okay. No. Ingression is different, Vijay. Ingression, which in which you have a layer of cells, and they are epithelial cells which are connected with some cell joints, cell to cell adhesion molecules. They are joined together. But some cells will lose the adhesions and they will come out as individual mesenchymal cells. These are epithelial layer of sheets. But some cells will come out as mesenchymal cells. That is what is called as ingression, that process. Okay. So that's why here the answer will be epiboly. See, these are the different kind of movements. During gastrulation, cell migrations take place. Epiboly is the movement of epithelial cells that spread as a unit to envelop a yolk mass or deeper layer. Remember in amphibian embryo gastrulation, I showed you the picture where the outer layer, ectodermal layer, was spreading over the entire embryo to cover the entire embryo, so that the outer layer is covering the embryo. Got it now, Vijay? Kirtana, got it? It is epiboly, right? So that is epiboly. Emboly is what? It is internalization. The same thing which we saw in amphibian embryo. How there is a creation of a blastopore. So cells are going inside. So basically it is internalization. That is emboly. It entails movements of mesodermal and endodermal precursors from the blastula surface beneath the ectodermal layer. So beneath the ectodermal layer, they will be going in. Involution is also interning or inward movement. Okay, that is your clue, because here it is saying it is covering, enclosing, covering deeper layers, means it is on the outside. Okay, involution, while involution is in the internal of an expanding outer layer, from there it will go inside. Inward movement of an outer layer to inside, that is involution. Yes, there also it will spread, but you have to see what they are doing. It is involution around the point or edge, that is inward movement. But here it is spread of an outside cell layer to cover a deeper layer. Okay. So that's why involution epiboly can be confusing. But here are your clues how to recognize the correct answer. And ingression, as you can see, these are individual cell movement. Whereas epiboly, involution, invagination, they are all migration of cells as a unit. Together they will migrate. Okay. But ingression is migration of individual cell from the surface layer into the interior of the embryo. So can you see here, these are the cells which have become independent. So they have come out as small groups from a epithelium sheet into the cavity of the embryo. Okay. So I want to remind you all, Shaurya batch for CSIR NET Life Sciences registrations. In fact, only limited seats are left. This is the last call to you. So if you want to be a part of it, you should enroll now. Classes have started. Rate classes by CSIR NET Expert. So it has been just a few classes. So interactive online classes will be there, doubt solving via chat, printed hard copy material, video backups of all classes, and basically you're getting a free video pen drive. So this is a good opportunity to avail the offer. So kindly contact us on the given numbers, either toll free or our Bangalore branch number, to get details of it if you want to clear CSIR for sure in the June session. Okay. Now coming to part C questions. First from the unit from shift 1. So this is about limb development. Okay. So can you answer this question? So a lot of questions were given from limb development these day. I mean these exam. Anyone wants to try? So as the limb grows outward, stylopod forms first, then zygopod, then autopod. And each phase is characterized by some specific expression of Hogg's gene. Then zone of polarizing activity is maintained by interaction of FGF from AER and SHH which are expressed from the mesenchyme. Right. So let's have a look. I haven't got any response, so probably you didn't attend this, but this was a very easy question. I mean, not easy, but yeah, if you at least know some logic about limb development. So basically the part which is proximal to the body, so we have our humerus, no, the top part of our arm, that is stylopod. The proximal part is the stylopod. Then what you have is the zygopod, the middle part, that is radius and ulna, what we have. And then the arm part, the hand part, the fingers and all that, that is the autopod. So remember that limb is coming out. So can you see proximal part and distal part? So limb is coming out like this, right? So which parts will form first? The proximal parts will form first, then the distal parts. So as the limb outgrows, yes, stylopod will be formed first, then zygopod, then autopod. This is correct statement. And these are controlled by expression of some Hogg's genes. We will come to that. In shift 2, there was a similar type of question. So there I'll tell you the Hogg's gene expression part. The next, it was given about ZPA. So basically, what is ZPA? So same diagram I'll show you. See, from here, this was the proximal part and this was the distal part, right? So same diagram, you can see this is the proximal distal. Now by the way, this is a limb bud. This is called as a limb bud. How flowers come from flower bud, like that limbs will outgrow from limb bud. Okay. Now in the outer part of the limb bud, can you see there is a thickening of the cells? Okay, a thickening layer of tissue is there. That tissue layer is called as apical ectodermal ridge. Yes, it is ectodermal tissue. That is why it is ectodermal. And ridge means what? It's a side area which is a thickened area. And apical means apex. Apex means at the tip. So this is at the tip of the limb, no? That's why apical ectodermal ridge. Then there is something which is just underneath it, and that's called as progress zone. Progress zone contains mesenchymal cells. Okay, progress zone will contain mesenchyme. Those mesenchyme cells which I showed you in ingression from epithelial layers, they were coming out to form individual cells, same types. So these are also mesenchymal cells. Basically, they are very much flexible. They can form any other type of cells also. Those are called as mesenchymal cells. Okay. So basically, uh, this is the posterior part and this is the anterior. I mean, this side is your head and this side is your legs. Okay. So at the posterior part, because there is a difference in your hand, the anterior part which is towards your head, that part looks different. The posterior part looks different. So how that anterior posterior differentiation is done? It is done by something called as ZPA, which is zone of polarizing activity. So how this is maintained, the zone of polarizing activity, it is maintained by the interaction of FGF8. So there is something called as FGF8 which is released from the AER. AER gives signal to progress zone gives signal to AER. This is a reciprocal signal which happens, okay? So basically the ZPA is established and maintained by this FGF8, but there is something which is only towards the posterior, okay? So that will come from the cells which are towards the posterior, which are made competent to express sonic hedgehog. So only when there is sonic hedgehog, and that depends on the respective Hogg's genes. So there are some layers, okay? There are some Hogg's gene layers Hox genes which are expressed. So wherever there is a specific Hox gene, that side only will be the posterior. A little before Hox genes, that side will be anterior. So like that, they get their signal. So can you see here SHH, how it is expressed in the area of zone of polarizing activity, right? And that zone of polarizing activity is giving signal to AER, where FGF4 and 8 is expressed. And AER is also giving signal to SHH to maintain and be called as ZPA, right? So second statement is correct because it says it is maintained by interaction of FGF from the AER and SHH which is expressed from the mesenchyme. Then the fourth statement, it says, so first of all, you have to select the incorrect statement. So 1 and 2 cannot be answered. Fourth is also right. Dorsal ventral axis is formed in part by expression of WNT7A in the dorsal portion of limb ectoderm. Yes, so even dorsal ventral has to be formed. That is, which is the back side of the hand, which is the front side of the hand. Again, there is a difference. So they are saying it is maintained by WNT7A, which is expressed in the dorsal portion. And that maintains expression level of SHH in the ZPA and FGF4 in the posterior AER. Okay, so let's see if this is correct. So they are saying the WNT7A maintains SHH in ZPA and FGF4 in posterior AER. So that's correct because you can see that WNT7A, it is expressed in the dorsal area, and how it is controlling through a signal called as LMX1B. There is a question regarding this also in shift 2 part C. So we'll discuss about that also. LMX1B, that is maintaining this SHH. Okay, and SHH is maintained by FGF8. So dorsal ventral axis is formed by expression of WNT7 in dorsal portion. It maintains the expression of SHH in ZPA, and it also maintains this. See this LMX1B, it is maintaining SHH also, and it is maintaining FGF8 expression in the posterior AER also. So fourth is also correct. However, third is not correct because cell death in the limb is necessary for the formation of digits and joints. Okay, and the effects of BMP can be regulated by the noggin protein, and BMPs can be involved both in inducing apoptosis and in differentiating the mesenchymal cells into cartilage. So BMPs are needed for the induction of apoptosis, right? Otherwise, for example, we have this initially in limb development, we have all our fingers as webbed, but later that webbing is gone. Webbed means how you have for the ducks and all that, no? So like that it would be there, but we don't have that webbing. How are those cells gone? They are gone by apoptosis, killing of cells in the um, killing of the cells in these areas where it is not necessary, and that is mediated by BMPs. Okay, that in turn is necessary, it is by noggin protein. So here it is saying although cell death in the limb is necessary for formation of joints, it is never mediated by BMP. So this is not correct since it is responsible. Okay, so answer is 3. 3 is the wrong answer. Okay, another question from part C in shift 1. This is regarding dorsal ventral formation, polarity formation in Drosophila, including the dorsal gherkin protein. Classic question which always comes, cactus, right? The following statements were made regarding the activity of dorsal protein in establishing the DV polarity. See, not necessarily you will always get mutation based question. Here they are simply asking some theories, some logics they are asking. So which of the above statements are correct? You have to choose any one of them. Did anyone answer this question? Are you following, students? Is it helping you? This discussion, is it helping you? Okay. So see if you can answer this question. You know this concept? Anyone aware of this concept? Do you want me to explain this concept? This is a very common topic of question, all asked almost in every question paper of CSIR, right? So yeah, basically what do we have here? See, the dorsal ventral polarity, it is established Just remember this part, by a gradient of a transcription factor called as dorsal. Only one point you have to remember. Dorsal, you might think that dorsal protein, if it is present, it will cause dorsalization. That is formation of dorsal part. But actually that is not correct. Okay. Dorsal protein causes ventralization because it is based on mutation phenotype. The naming is done on that. That if dorsal is not present, then it will not cause ventralization. Not causing ventralization means it is causing dorsalization. So it is based on mutational phenotype that if it is not there, what will happen. Okay. So dorsal causes ventralization. This part we understood. Now what happens? I'll show you a diagram. Basically, this is the Drosophila embryo. You have some nerve cells. Then this is yellow color is the embryo with the nucleus. Actually, this is the egg. This is just the egg. It has not yet formed an embryo. Okay. And these nearby cells, what you see, these are follicle cells. Okay. So there are some dorsal side follicle cells. There are some ventral side follicle cells. So before fertilization only, their specification is done. Which follicle cells are going to take dorsal fate? Which follicle cells are going to take ventral fate? Now, how is this specification done? It is by the activation of the pipe protein synthesis. So remember, in the dorsal side, which side is supposed to form dorsal, pipe protein is inhibited. It is not being synthesized. Whereas on the ventral side, prospective ventral side, pipe protein will be synthesized. Okay, I'm just telling you the gist of the story. Now, if pipe protein is there, then what happens? We have to connect it with the dorsal protein, right? So just remember, if pipe protein is there, okay, there is a signaling cascade, not going into detail. If pipe protein is there, it degrades cactus. See how cactus is degraded? Now, cactus was supposed to bind dorsal. Now, what if cactus is degraded? Dorsal is free, right? So, dorsal can enter nucleus. And if dorsal enters nucleus, I already told you, it leads to ventral phenotype. Okay? Now, let's understand what is happening at the dorsal side. We don't have pipe protein. If pipe protein is not there, then it cannot degrade cactus. If it cannot degrade cactus, dorsal will still be bonded. And if dorsal is bonded, it cannot enter nucleus. If dorsal cannot enter nucleus, it will lead to dorsalization. That's why this side forms dorsal fate. And because dorsal enters the follicle cells, means dorsal enters the nucleus on this side, that is why they will take the ventral fate. Okay. So while dorsal protein can be found throughout the syncytial blastoderm of an early Drosophila embryo, dorsal is present all over. But later what happens when the embryo will get fertilized? So it will make many cells, right? Right now it is in the egg form, but later it will make many cells. So basically, if dorsal is present free over this side, then it will enter the nucleus of all these cells. But over in the dorsal side, it will not enter the nucleus. So the cells which it doesn't enter the nucleus, those cells will form the dorsal cells, and the ones where they enter the nucleus will form the ventral cells. Okay. So if dorsal is found throughout entire embryo, but at the dorsal side it is bound by cactus. That's why it cannot enter nucleus. So it is translocated into nucleus only in ventral part where it is free, because pipe protein has degraded cactus. That's why dorsal was free. Okay. Now, what is it doing in the nucleus? Why the dorsal is giving ventralization? Because it binds to certain genes either to activate or suppress their transcription. So, what did I tell you? That dorsal will make ventral genes. So, if dorsal enters nucleus, it will make ventral cell types. But if dorsal does not enter nucleus, the genes which was responsible for making ventral, they won't be made, no, because dorsal was making ventral. So in that case, what will happen? The genes which will specify dorsal, they will get expressed. So they are not repressed because dorsal was basically repressing them and activating ventral. But if dorsal is not there, then dorsal cell types will be transcribed. Okay. Now cactus, I told you, it blocks the portion of the dorsal protein that enables protein to nuclei. So there is a gradient of dorsal translocation in the ventral cells with the highest concentration in the ventral cell nuclei. Now, if cactus is not present, cactus itself is not present, then no need of entire signaling, no. By then this embryo is special in that cactus is not present. Then dorsal will be present free all over. This side also dorsal will be there, this side also dorsal will be there. So entire nucleus will become ventral because dorsal is entering nucleus of both the sides. Now let's see if we can answer this question. So embryo that lack gherkin protein, the dorsal protein is not translocated. Okay, that was the first statement. Sorry, yeah. So in embryos that lack gherkin, yeah, I did not mention this to you how the pipe protein is synthesized. So pipe protein is synthesized in the ventral side because there is no gherkin mRNA. And why pipe protein is inhibited in the dorsal side? Because there is gherkin mRNA. Okay, can you see the nucleus slightly towards the dorsal side? That is why only in the dorsal side you will have this gherkin mRNA, which will bind to the torpedo receptors on the follicle cells. And that is how the pipe protein won't be synthesized in the dorsal side. But pipe protein will be synthesized this side because there is no gherkin and follicle cells won't be activated. That means torpedo won't be activated. So pipe protein synthesis will not be inhibited, means it will be synthesized. So gherkin, if it is not there, then pipe will get synthesized both sides, the dorsal side also and the ventral side also. Okay, but what is it saying? That the dorsal protein is not translocated to the nucleus, which then causes ventralization of the embryo. No, because if dorsal is translocated, it will cause ventralization. If it is not translocated, it will cause dorsalization. So this is wrong. Though dorsal protein acts as a morphogen, it is found throughout the syncytial blastulum of the early Drosophila embryo. So this is correct as we saw. And definitely it is acting as a morphogen as it is specifying dorsal and ventral fates. In embryos that lack cactus protein, dorsal protein can be found in nucleus of cells with a ventral fate. Yes, because if cactus is not there, then dorsal protein is free everywhere, in dorsal side also, in ventral side also. So it can be found in the nucleus of cells and they all will give a ventral fate. If dorsal is blocked from entering nucleus, then will be no ventralization. In that case, the genes responsible for specifying dorsal cells are not transcribed. No, they will be transcribed because dorsal is blocking or repressing the dorsal cell types specification. So if dorsal is blocked from entering nucleus, nobody can repress the dorsal genes, so they will be synthesized. That's why D is wrong. Here your correct answer is option 2, B and C. This requires a pretty good concept from classes. Here I told the entire concept in 5 minutes or so, but in classes this entire topic is discussed through one class. An entire class goes on this understanding and solving lot of related questions from here. Okay? So that's how classes make a difference. I'm sure those who have attended this topic class, they would have attended and got this question correct. Next question. Match the following. So again from shift 1, again it's a vertebrate limb development. As I told, many questions were there. They have given some terms: EMT, mesenchyme, AER, progress zone, ZPA, autopod. Somehow you know few terms from here from today's session. So can you attempt to answer this question now? Let's see. Okay. Yeah, that's right, Amit. B and C for previous question. So Bhavya, are you answering for this question or previous question? Okay. So I have got answer from Kirtana and Vijay. So you all know what is autopod, right? What is autopod? I just told you in some previous question, stylopod, zygopod and autopod. So can you all answer what is autopod? I hope you have been paying attention. So it's a distal part, isn't it? Yes. That means any out of these three will be correct. Okay, so that didn't help us. We will select another one. What else you know? Probably you will know apical ectodermal ridge. Okay. Okay, let's see, Amit. So probably you will know apical ectodermal ridge. So I have told you that it's a thickening area. That too at the epidermal, that is ectodermal part, at the apex. If you know apical ectodermal ridge, so C will be 2. So let's see if it helped us. Yes, C is 2. Yes, it did help us. Option 1 is the right answer. So see, only with two concepts I have solved this question, and I have chosen the easiest ones, isn't it? So with that only the question is solved. C is 2. So what else is others? What is EMT? Epithelial mesenchyme transition. So if you don't know that, anyways I told you by the process of ingression they will do that. Epithelial cells making up the mesoderm of the early somatoplure undergo this transition and get included in mesenchymal cell pool, because epithelial cells will change their cells, means they will come out from the layer of cells through the process of ingression and become a part of mesenchyme. What are mesenchyme? That also I mentioned, that they are some cells which are loosely present, not attached in a cell, and they will be mostly making mesodermal cells of the embryo later. Progress zone, I told you AER is there. Just below AER, progress zone cells are present. So basically, they will proliferate. They are mesenchymal cells only which are proliferative. They will proliferate so that the limb outgrowth will take place. Limb has to grow, right, from away from the body. That outgrowth is maintained by proliferative progress zone. And zone of polarizing activity, I mentioned that it is present at the posterior area of the limb, which gives posteriorization. So it's cells found within the most posterior region of the limb bud. Yes, Amit, that's right. So correct option is answer 1. No need to explain anything. Everything I just mentioned to you. So basically this is the apical ectodermal ridge. This is progress zone. This is ZPA. And this is stylopod, zygopod. Autopod is most distal part. And yes, this is apical ectodermal ridge. This is ZPA. And these are all mesenchymal cells. Okay. So answer 1 is correct. Okay. Another part C question was there in shift 1 about neurulation. Primary neurulation, secondary neurulation. So which of the following options gives all correct statements? So can you answer this? Did anyone opt for this question? So they have asked you about primary neurulation process, secondary neurulation process, plus they have asked what happens in birds, what happens in mammals. Like from where primary neurulation, till what primary neurulation will be there, till what point secondary neurulation will be there. And they have also given about a condition, anencephaly, and they have said what is the reason for that. Means what happens because of anencephaly. Okay, no problem, Amit. We will see about that. Results when a failure to close the neural tube will occur, resulting in forebrain remaining in contact with amniotic fluid. So definitely there will be a, a difference what will happen over there, right? So these all topics they come under what we discuss in classes. That's why this is again a reminder. It's a last call for Shaurya batch. Only limited few seats are left. We have extended based upon request. So if anyone is interested, then you should call and contact us now itself before the gets closed. Okay. And that too we teach with RAID methodology. It's a new type of methodology of teaching in class, which Biotecnika has found, and we are going to conduct a webinar on that. That is upcoming on 15th March. Book your dates, book your calendar for the dates. So we'll be telling you how exactly this rate classes, what's the new concept of this rate classes. Okay, coming to the question, primary and secondary neurulation. Let me show you the process. There are two major ways of forming a neural tube. So in primary neurulation, what happens? The cells surrounding the neural plate direct the neural plate cells to proliferate. So basically, this is the back side, okay? That is the dorsal side. So what happens? So this is mammal. They have asked what happens in mammals and birds. This is mammals. So see, till somewhere it is primary, then there is a transition zone, then from somewhere it is secondary neural tube. So the process of formation is different, that's all. What happens in primary? The epithelial cell only makes a neural plate by columnarization that undergoes rolling or folding, makes a tube like structure, closes, and finally it forms neural tube. So basically, primary neural tube comes from epithelium. But secondary neural tube, the neural tube arises from a solid cord of cells that sinks into the embryo and subsequently hollows out. So they are dispersed mesenchyme. They are actually aggregation of mesenchymal cells, which are cord or neural rod, and then there is a cavitation which is built inside, because of which cavity develops inside. Thus, it forms a tube-like structure. Okay, so that's correct. A and B both are correct. In primary neurulation, the cells surrounding neural plate direct the neural plate cells to proliferate, invaginate, separate from the surface ectoderm to form an underlying hollow tube. And in secondary, it arises from an aggregation of mesenchyme cells into a solid cord that subsequently form cavities to create a hollow tube. Now, if you see, I told both A and B are correct. So that means option 1 is right answer. Yes, the rest all are correct. In birds, it arises the neural tube from anterior tip up to the hind limb developing region. Okay, that is primary neurulation is up to some mark. In secondary in mammals, primary neurulation is up till the sacral vertebrae. Beyond that, it begins from the level of sacral vertebrae, secondary neurulation. So in birds, the anterior portion of neural tube are constructed by primary, while the neural tube caudal to 27 somites. Somites are some levels in the vertebrate body. Okay, some levels of mesenchymal type of cells which are necessary. Everything posterior to the hind limb is made by secondary neurulation. In mice, neurulation in embryos starts below the level of 32nd to 34th somite, which corresponds to future 3rd to 5th sacral vertebrae. And yes, the last was condition anencephaly, which is also correct. Failure to close the anterior neural tube regions result in a lethal condition. Here, the forebrain remains in contact with the amniotic fluid and subsequently degenerates. Fetal brain development is also affected and the skulls fail to form. Okay, so E is also right. So, yes, I want you to remember miracles start to happen when you give as much energy to your dreams as much as you do to your fears. So, overcome your fears, be a part of Biotecnika classes, and get guaranteed success. Okay, coming to part C questions. So, this was question 239. Yes, the part C shift to questions were actually very conceptual. Okay, if you don't attend classes, then it's quite difficult to get the concept all by yourself. Means you have to do a lot of hard work. You need a lot of dedicated time to study from books and all that. But yes, since in classes you get all streamlined information, so definitely all this you can get very easily. Okay, so in an experiment, there is a molecule called as activin. Secreting beads were placed in unspecified cells from an early xenopus embryo. Okay, so activin then diffused from the beads. So if beads contained 1 millimolar of activin, it elicited expression of XBRA gene in cells near to the beads. Now, if the beads contain 4 nanomolar activin, okay, if it contain 1 nanomolar, then the cells which are near to the bead, consider this is the bead. So cells which are near to the bead, they expressed XBRA. But that was with 1 nanomolar. So again, if you would have noticed, I have drawn a diagram, isn't it? 4 nanomolar, more you have taken. So the nearby cells, the expression of XBRA cells was elicited in cells, but only at a distance. Means the nearby cells, there is no XBRA, but in cells little farther away, there is XBRA expression. There you have XBRA expression. Okay. In the later case, that means in this case, the expression of goosecoid gene was observed near the source bead. That means in these cells where XBRA was not expressed, there we saw goosecoid. Okay. So we understand there is some link with XBRA and goosecoid. Something is inhibiting something. That's why they couldn't express. Okay. Now, beads with no activin, this is a control, did not elicit expression of either of the two genes, neither XBRA nor goosecoid. Now, following statements were made. High concentration of activin activates goosecoid, whereas lower concentration activate XBRA. So that is quite obvious. This is obvious over here. We have to select which are correct. Because the cells which were near to it, their goosecoid was activated, but with lower concentration, it activated XBRA. Lower concentration of activin helped specify dorsal most structures. Now, this is something which you have to know, that if you have less concentration of XBRA, then does it give dorsal structures or not? Then another thing you have to know is concentration of activin that do not lead to expression of two genes specify cells to become blood vessels and heart. So these are more ventral structures. Means they are asking if you have very, very less concentration of activin, then that becomes the ventral part. And they are asking lower concentration helps specify dorsal. So basically, either of these two statements should be correct, right? Either A and B or A or C. So let's see which is correct over here, right? So correct answer over here is option 3. I told you either A and C will be correct or A and B will be correct. So there is no option of A and B. So that's why A and C, because yes, lower concentration of activin means, see, if higher concentration of activin is there, it will be activating goosecoid. If you remember, I showed you in the first question, I told you you will require this concept. Goosecoid was giving dorsalization, right? So high concentration of activin will give dorsalization, but it is saying lower concentration helps specify dorsal most. That's not correct. Okay, whereas lower concentration of activin, there you will have the ventral phenotype where cells will become vessels and heart. So basically I've given you the concept over here, the same thing which is written in the question. What happened in 1 nanomolar, this was the experiment which they did. Okay, so this is the activin bead kept in the center. Here there is no activin bead. Okay, and this is 2 hours post. This is not the same experiment, but yeah, these two experiments are correct. If you have more activin, then XBRA is expressed on the outside and goosecoid will be expressed inside. The reason is because they inhibit each other. So activin high expression is going to give goosecoid. Low activin is going to give XBRA. But if it is beyond the threshold concentration, neither of this will be activated. That is why in the control experiment, neither of these genes were there. And if XBRA is present, it is going to inhibit goosecoid. If goosecoid is present, it is going to inhibit XBRA. So since in low concentration of activin, you have XBRA, so that's why in high concentration, when you took 4 nanomolar, here you did not get XBRA. So if XBRA is not there, goosecoid is activated in high concentration. If goosecoid is there, it is inhibiting XBRA. When you took 1 nanomolar, so immediately in the nearby cells, they got low concentration. So that's why they formed XBRA. Because they formed XBRA, goosecoid could not be expressed. That's why you saw this kind of observation. Okay. So since 1 nanomolar is low concentration, less activin was diffused in the nearby cells. Also in 4 nanomolar concentration, distant cells would get less activin when it diffuses from source bead. Thus, only the distant cells could activate XBRA and not the nearby cells, which received high activin concentration. So we understood that goosecoid inhibits XBRA. Hence, it can be concluded that in xenopus embryo, activin induces a range of mesodermal genes in a concentration dependent manner. The genes XBRA and goosecoid are activated in low and high respectively. So that's why A is correct. Vertebrate homeobox gene goosecoid is involved in dorsal ventral patterning of mesoderm and is expressed in dorsal lip of blastopore tissue. So thus low concentration of activin will not express goosecoid and will not lead to formation of dorsal node structure. And when the animal caps are treated with extremely less activin, so basically they don't get activin signaling. Because of that, they won't secrete any of these dorsalizing factors. So then they differentiate into ventral mesoderm, which includes mesenchyme and hematopoietic cells, the ones which will give rise to blood cells. So thus, if activin concentration is so less that neither of the genes are expressed, then it forms blood cells and mesenchyme. So C is also correct. Okay, option 3 is the right answer. Next question was about sea urchin. Yes, this was also a amazing question. Okay, from a question point of view, it is really amazing. I don't know how you will feel as students to be answering, but yes, this was a really good question. The specification of sea urchin micromeal involves the activation of a repressor protein, PMAR. I have just told you sea urchin in one of the previous questions, also in part B. So basically, this is shown from the bottom. Basically, these are micromeres. And you can see that in these micromeres, we know that beta catenin is expressed, right? So basically, what happens with this beta catenin, that is regarding the question, right? Now, this specification of the micromere involves activation of a PMAR protein. So, I will tell you over here, beta cat will activate PMAR. Wherever it is present, beta cat, that is in micromeres, PMAR is activated. PMAR inhibits a protein because it is said it's a repressor protein. It represses the expression of HESC. It's written also in the question. It inhibits HESC, which also encodes a repressor protein. Okay, so this also will repress something. One of the genes which HESC represses is delta, right? So that delta protein specifies micromeres. Now that makes sense. This is a double inhibition pathway. You can see double negative pathway, right? Now in micromeres, beta cat present. Because of that, PMAR1 present. PMAR1 present means HESC not present because it is inhibiting HESC. HESC not present means delta present. Delta present means micromere specified. Okay. So they have done an experiment, that is whole mount in situ hybridization, WMISH, to find the delta probe. That means basically something which will detect delta. Now, this is a normal experiment where you can see the delta is expressed only in the micromere regions. And you know why? Because of beta catenin, because of PMAR, because of inhibition of HESC, and because of expression of delta. Okay. The rest of the embryo, we will not see delta because there beta catenin is not there. So if beta catenin is not there, PMAR won't be there. PMAR is not there means HESC will be present. PMAR was supposed to repress HESC. If PMAR is not present, then HESC will be present. And if HESC is present, it will inhibit delta. So delta you can't see in the rest of the places. You have to make the correct match. This is the actual question. So what happens where you will find these following things? If you do, this is an experiment to find out delta. The colored portion is delta. Okay. But if you do a WMISH with HESC probe, then what will happen? Where it will be expressed? Either in A or only in B or both A and B or neither A and B. Like this, different they have given concepts. So just imagine how much concept you require to solve this question, right? So let us see. Now I told you that HESC, if it is present, HESC if it is present, then we will not see delta, right? But when is HESC present? They have not given any condition. They have just said HESC if it is present or not. Okay, we have tried to say, no, you have to match, no? So I haven't given you the options yet. These are the options. You have to get one or two. You have to match column A with column B, Amit. So if you do that, WMISH with HESC probe, then where will you see the expression? Okay, that color will appear where? If you do WMISH for delta probe, that means the color will only be present where delta is getting expressed. Got it, Amit? Where delta is getting expressed, there only you're getting color. Same if you do WMISH with HESC probe, you'll get color only where HESC is present. Now, where HESC is present, where PMAR1 is not present, and where PMAR1 will not be present in the rest of the cells because there beta catenin won't be there, no? Okay. So that's why, okay, let's see Kirtana. So that's why we will see WMISH HESC probe only in the rest of the embryo, that is in B area, because there HESC will be present. Okay. So A will be 2. Got it? So A is 2 means either 1 or 2 is right. Got it? Let's see B now. Now WMISH with delta probe. You want to get delta only. like this experiment, but you have a condition. What have you done? You have injected PMAR1 into fertilized egg. Now, where was PMAR1 getting expressed? Only in this area, naturally. But you have done an experiment where you have put PMAR everywhere. Into the egg itself, you have put PMAR. So it will be present everywhere. Okay, initially it was supposed to be present only here, because of which you got delta here. But now it is present everywhere. So everywhere you will get delta. Everywhere it will be colored because you are doing with delta probe. So both A and B. B will be 3. Okay, so B is 3. That means again you have the same answer. A, B, okay, any of these. So see, this was a really difficult question because even after solving 2, you could not get the answer. So the last one, W mesh with delta probe following. Again, you are doing for W mesh. Sorry for that. Again, you are doing for delta probe only. Micro injection of antisense RNA to HESC. So antisense to HESC means what? HESC won't be there. Yeah, that's what, Amit. Antisense means HESC won't be there. The HESC mRNA will be there, right? Antisense mRNA will go and bind with the actual mRNA, so HESC won't be present. Now, HESC was supposed to be present where? First of all, you have micro-injected that antisense mRNA everywhere in the entire fertilized embryo. So, HESC was present earlier in entire rest of the embryo, except this area, right? So, except this area, if it is present, then the rest of the ones, but what you have done, there also you have put antisense mRNA. Means where it was supposed to be present, there also now it is not present. And then you have done with delta probe. So delta probe means what? If HESC is not there, then delta will be present here also. Correct? So here, that's why the correct answer will be option 1. Got it, Kirtana? Because see, microinjection of antisense mRNA means HESC not there. So here where HESC was supposed to be present, there also it is not there. And if HESC is not there, delta will be there, no? And if delta is there, then color will be here also. So C will also be 3, both A and B. I understand. By just knowing the concept, it's very difficult. And that too if you're learning the concept for the first time. That's why classes help. If you know these concepts in classes with practice, because all this have already been discussed, not this exact experiment, but yes, this concept is there, and we do other question practices also, which makes the concept even more clear. That concept which I just explained, it is given in a flowchart form. You can refer it later with the explanation also if anything is not clear. Okay. Another question from part C regarding induction. Shift 2. It is an extrinsic process that depends on the position of a cell in the embryo. It is a process whereby one group of cell can influence the development fate of another, and it's a common strategy to control differentiation and pattern formation. One cell giving signal to another, that's how the cell's induction will be formed. So following statements were made. Inductive signal can be a protein secreted from the inducing cell that binds to receptors of responding cell. Response to inductive signal depends on competence of the inducing cell. Instructive interaction occurs when responding cell is already committed to a certain fate. Lateral inhibition results in differentiation of individual cells in a regularly spaced pattern. Which out of the statements are correct? So you know cell signaling, how the induction and response works. So you might be able to figure this out. So as we know that for an induction, there are at least two components to every interaction, right? The first component is the inducer. So the first component is the inducer, the cell which is giving the signal. And the second component is the responder. So inducer is the one which is producing the signal, which will change the cellular behavior of the other tissue. Second component, okay, let's see Kirtana, is the one which is getting induced. So this is the responder. Now every cell won't be able to take the signal, okay? Only cells which have competence, they will be able to take the signal. But the thing is, they have said that it should have competence. But what have they said? Inducing cell should have competence. No, responding cell should have competence. Simple logic of competence means capacity to respond. Ability to respond to a specific inductive signal is what is competence. But that is a property for the responding cell, not the inductive cell. That's why statement B was wrong. Okay. In instructive interaction, and first statement was correct because you have an inducer and responder. In instructive interaction, one cell will give new signals to the responding cell. Without this inducing cell, this responding cell won't be able to form a particular type of tissue. That is what is instructive. It is giving instruction. Okay, but it is saying that, yeah, the instruction is for initiating new gene expression. But see what was it saying, that in instructive interaction, it is given to cells which are already committed. That's not correct. If they're already committed, means they would have already got instruction. But this is for getting new gene expression. So C is wrong. Tissue organization and patterning are critical during development when genetically identical cells take on different fates. So lateral signaling means what? That is the inhibition. So inductive signal, I already mentioned you, one cell giving signal. And then another cell will give different signal to the nearby cells. So see, even if all these cells are getting signal, but because of lateral inhibition, lateral means sideways inhibition, they form something else. So can you see a pattern has formed? A dark green color showing different pattern, light green color showing different pattern. They did not get signal, so different pattern. That's how it plays important role in process by helping to generate self-organized spatial patterns in an otherwise uniform collection of cells. Initially, these cells all were same, but because of the combination of instruction and lateral inhibitions, they found something else. That's why this is correct, these are wrong, and D is right. Third is the right option. Okay. All right. Then the last question, I think second last. Given below are statements related to expression of some of the pattern forming genes in vertebrate limb bud. That LMB1X gene is expressed in dorsal. We saw this today. I hope you remember what activated LMB1X. So there was a WNT7A gene which is expressed in dorsal ectoderm, right? SHH, whether it is expressed in posterior. HOXA13 and HOXD13 expressed in distal region. TBX5 FGF10 expressed in lateral plate mesoderm, involved in formation of limb bud. What many students would have done, directly they will start picking up true or false from here. But that's not the question. They have asked about a syndrome, nail patella syndrome, and they are asking in human and the syndrome in mouse where foot pads on both dorsal and ventral side will be there. They are associated with which of the following gene function? These have just given some gene functions. All of these gene functions are correct. But the question was nail patella syndrome will be seen because of which mutation, like which associated with mutation in which of this gene function, associated with the problem in which of this gene function. Okay. So here the correct answer, if you don't know, probably many of you won't have heard of this nail patella syndrome. It is LMBX1B and WNT7A. So both are expressed in dorsal ectoderm. The LMB homeodomain transcription factor is responsible for limb dorsalization. So it is limb 1x. I told you it is same diagram I have kept. It is activated by it is induced by WNT7A from the dorsal ectoderm, and its expression is restricted to the dorsal side only. So if there is loss of this LMBX1B function, then it results in loss of any of these. Dorsal autopod won't form, zygopod won't form, patterning won't form, and autopods will have a symmetrical ventral to ventral phenotype. Instead of dorsal to dorsal, they have just ventral to ventral. So that will result in dorsal foot pads. So absence of nails and all that will happen, and that results in this syndrome. Okay. Besides the limb mice lacking functional LMB and all that. And I just want to show you this proximal distal part. So TBX, I told you, no, Hox gene controls. So you can see that 13, Hox 13, it controls the forelimb in hind limb, the distal parts. That's what the Hox 13 will control. So how it is written, no, that is Hox 13 and Hox D13. They are expressed in the distal region. So that's what I wanted to mention. Okay, option 4 is right. This is the last question in shift 2. This is regarding the amphibian and avian, means birds, chicks, and amphibian. Some terms are given from amphibian gastrulation, some terms are given from avian gastrulation. You have to match similar roles, similar terms. That means which have similar functions. Okay. So, what will be the answer? For example, what is the role of nucleus center in amphibian? Which part in avian will have a similar role? Like that, you have to match. So, it's a again conceptual question, but not that difficult. Anyone who has attended Biotecnika classes will easily solve this question, because while studying different development, we do compare. For example, amphibian and avian gastrulation is something which we compare all the time. Okay, what is the difference? What is the similarity? And here we have a question based on that. Yeah. So basically, what do we know? What is blastocele? So in amphibian development, you have a blastocele which is formed over here, right? And in the avian development, basically avian development, the blastula, actually it's a blastoderm which is only in the topmost part, because the rest of the cells, they are filled up with yolk. So no development over there, means no division, cell division will take place only in the anterior most tip. Okay, a group of cells. So there one upper layer will be there which is called as epiblast. Okay, and then a middle layer, so this is the egg from the side, then there will be a bottom layer will be called as hypoblast. In between epiblast and hypoblast, that space will be blastocele. So A will be 4. Okay, so A4 we have either 1 or 2 is correct. Okay, then let's see blastopore lip. You know there is a blastopore lip which starts from here, where the movement of the cells will start. The cells will start to go inside. Okay, so inside means sorry, yeah, so inside means the cells will start to migrate, and that happens, see, basically in the epiblast, a line is formed, a streak is formed which is called as primitive streak. Okay, a line is formed, primitive streak. In that primitive streak only the cells will start to migrate, means through that streak, it's an opening, you can say, through that streak the cells will start going inside. So B has to be, blastopore lip has to be 2. Yes, you got your answer. Dorsal lip of blastopore, the cells which are present near the blastopore lip. So the cells which will go inside, that is the Henson's node. Henson's node is the first place from where the cells will actually start to go inside. Okay. Blastopore, yeah, it is called as the gap. What you say, that gap is the primitive groove. Groove is the part where, yeah, correct, Amit, option 2. So primitive groove is the part where it's an opening inside. So D will be 5. And nucleus center, we all know what was nucleus center doing, right? It was originating the organizer. It was giving signal to organizer. So yes, it is equivalent to the posterior marginal zone cells in amphibians. So here is a uh, comparison of the two things, right? So at the anterior end of primitive streak, so as you can see from the top, this is how the streak forms. Streak elongates. Streak forms further, elongating. Okay. And if you see from a cut section, then you're able to see, yes, this is epiblast, this is hypoblast. In between, there will be the blastocele. You can see in between, there is the blastocele between the epiblast and the hypoblast. Okay. And Henson's node is the place where is the, which is acting as the tip portion, dorsal lip of blastopore compared to that. Okay. So it acts as the equivalent of the PMZ acts as the equivalent of Henson's node. So that's all. So if you're interested in Shaurya batch, do call us now. That's all for now. Thank you everyone for joining in. We'll be back with another session for Unit 6. Thank you. Bye.